Thanks for coming. So it's a great pleasure and a great honor to introduce Professor Adam Chippindale. For those of us who work in the field of evolutionary biology, we don't really need to introduce him. But given that a few of you are not from this field, I will quickly tell you about Adam. So Adam did his PhD with Professor Michael Rose at UC Irvine, a very big name in the field of aging research in particular, and Drosophila life history evolution in general. Then after that, he went to, sorry, you can't listen? Oh, great. So after that, he went and did his postdoc with Professor Bill Rice, and that's when he got interested in sexual conflict. And then since 2002, Adam has been at Queen's University, Canada. And he initially did a lot of work on sexual selection, sexual conflict, etc. And then of late, he has become interested again in the evolution of aging. And just this afternoon, I learned that currently he is writing a book on menopause, a popular science book on menopause. But today, he is going to talk about sexual conflict and in the multiple ways in which it manifests and the multiple ways in which it affects. And yeah, that's about it. So, Adam, over to you.
Thank you very much, Suchi. Thank you for that kind introduction. I'm super excited. I'm really delighted. I'm honored, maybe a little jet lagged, to be here in India, to be here as a guest of INSA, to be here as a guest of IISER Pune. It's really exciting. I have seen so many old friends already just in a few days and made so many new ones and so many really vibrant scientists, especially young ones. Hanging out with them has been a real treat. So I have a few more days left and I'm expecting more of the same. What I'm going to tell you about today actually is the story of 3 decades of research. I'm going to tell it in 3 chapters, and I'm going to tell you about 3 amazing Indian scientists that have contributed to my research program since I set up shop in Kingston, Ontario, 23 years ago. Okay, the talk is about sexual conflict. I'm going to talk at first a little bit about what we might call an arms race between the sexes, or evolutionary chase, a kind of conflict that was really developed at the time that I was a PhD student in the mid-1990s. The papers are coming out fast and furious. Papers from people like Jeff Parker, Joran Arnquist and Locke Rowe, Bill Rice, Tracy Chapman, and you can see some of the organisms here, all insects, yellow dung flies, water striders or pond skaters, depending on your predilection, and of course fruit flies. And the idea of this kind of sexual conflict is that the sexes are inevitably going to be involved, engaged, connected. They have to be in some kind of interactions over sexual reproduction. And because they have different interests, potentially, in sexual reproduction, potentially different best strategies for maximizing their Darwinian fitness, that puts them into conflict. So a lot of this early work was on direct conflicts. Yellow dung flies scrapping and struggling for mates. Water striders in conflict over mating. Just let me give you an example of this kind of conflict. Okay, so there we go with the idea of the arms race. We have one sex adapting to the other sex adapting back. A lot of that 80s and 90s vintage research involves the idea that males are the active sex and females are the reactive sex, and I'm going to show you that that whole conception has been turned on its head. Okay, whoops. So back to the water striders. This is a group where sexual conflict is very flagrant, and it's a wonderful group to study, partly because a lot of the action happens in 2 dimensions. So people work on water striders in large part, not just for the cool biology, but because they spend their lives in a very easily traceable environment that is 2 dimensions moving about on the water surface. The sex lives of water striders can be incredibly brutal. Here's an example rich in morphology, some wonderful electron micrographs on the next slide from Ahab Abuhif's lab. Ahab here is shown giving a lecture at Harvard University. He's from McGill University, or he was until very recently. He's wearing a t-shirt that says, Harvard, America's McGill. That's cheeky, okay? So take a look at this development, this ontogeny of the antenna in this species of Jared waterstrider. So in this species, you can see the development of the female on the bottom, relatively normal giantization of a structure and very little change through metamorphosis. But when you look at the male, you can see that the same humble-looking antenna undergoes a massive metamorphosis in the last mold. Going into adulthood, it changes into what looks like a medieval torture device. And each of these things, these spikes and hooks and prickles and things like that, has a function that Abu Heath and company have quantified. Have looked at, for example, through electron micrography. Here you can see some correspondence between the parts of the male and the parts of the female that they're designed to attach to. So this is a grappling organ. These animals participate in violent pre-mating struggles. This exemplifies a frequent conflict between the sexes, and that's that males are often more interested in mating than females. And so the classic dichotomy between the sexes is that because males make microgametes, they can fertilize multiple females, their reproduction is limited by access to mates, and females' reproduction is often limited by access to resources and not fertilizing sperm. This leads to different strategies in a large number of species. Those sex roles, although they can be reversed, of course, especially when males contribute to parenting. Okay, so take a look at those structures. The correspondence is extraordinary, and the whole point here is to get yourself gouged into and around the female's eyeballs in order to gain a foothold, a grappling point in this battle between the sexes over reproduction. Okay, but as I said, females can play this game too. In water striders, females often outsize males, so they're bigger than males and they can repulse them if they try to mate. They often have slippery elytra, those are the wing covers on the back, that helps to shed males if they don't want them to mate. They often have defensive spines. But here's an example of a single species that was discovered by these two researchers, Chang Han and Pyotr Jablonsky, in a single lake in South Korea, a water strider in which females have an unusual genitalic morphology that is, as they put it, concealed. So here is a look at those concealed genitalia. The female's genitalia are covered up in such a way that the male can't mate with her. Even if he successfully wins the pre-mating struggle, he can't mate with the female unless she opens up this drawbridge-like apparatus and extrudes her genitalia. It would seem that even if the male wins the battle of the sexes in terms of the pre-mating struggle, the female now has the upper hand in the next phase. And you can see that Geras gracilis, the species I'm referring to, is unique in this group of Jared water striders in having this kind of morphology where the female genitalia are concealed. And this leads to a kind of mating that they characterized as being P2, which basically means that there's a second phase of courtship. So here is the second phase. Males, if they do gain amplexus with the female, if they do get into the mating position, the female is not going to open up that drawbridge and allow the male to mate unless he sings to her again. So this is what Han and Jablonsky called intimate courtship. The male has to court again in order to gain mating, and the female is in charge. That's the idea. Clever experiment. They put on the female something they call the W bar. It's just a little wire harness that prevents the male from tapping on the water. And you can see from this figure that it takes a whole lot more time and energy for the male to achieve copulation, if in fact he does at all, compared to controls where there's no W bar or there's a sham manipulation that simulates the attachment procedure without occluding the mating, the courtship drumming. Okay, so it's a neat story and it seems like females are in charge. So we can flip the tables here and say now the females are on top. Now the females are the active sex and the males have to come up with a solution to this problem of the females being in control. Well, this is an interesting story. I got involved in this. I was actually the editor on the first paper that they submitted, and I speculated to the authors that, maybe this is not just about courtship. This is not just about love matters, maybe there's some kind of sexual conflict involved, and in fact, maybe males are doing something else. And maybe they're trying to intimidate the females. And the authors rejected my suggestion, but it was almost exactly 12 months later when they published a second paper in the same series, suggesting that actually predators might be kind of important in the system. Here's why. So here's the paper. Male water striders attract predators to intimidate females into copulation, was the title. They used hemipterans, these are back swimmers. These make a living finding prey by swimming upside down underneath the water's surface, looking for things like struggling insects on the surface of the water. Hemipterans, fish, underwater predators are going to be attracted to things like insects drumming on the water's surface, sounding like an insect flailing, a perfect prey item, and probably saying to themselves, a 2-for-1 special going on here. And guess who's on the bottom? The female. So Hannah and Jablonski conclude that maybe this isn't sexual selection at all. Maybe this is sexual conflict. Maybe this is a form of terrorism, scaring the female into copulation. Okay, my story was introduced briefly. I was a PhD student in Michael Rose's lab in Southern California at UC Irvine. Was working on the evolution of aging, the evolution of life histories more broadly, and it became increasingly interesting to me that the sexes might be in conflict with one another. I was reading this literature, but I was thinking about a different kind of conflict. So I was thinking about the other kind I mentioned at the beginning, the arms wrestle rather than the arms race, the tug of war between the sexes, the problem of the sexes sharing a common genome. Okay, I still went to work with Bill Rice, and that was the idea, was to work on sexual conflict of the second type. At the time, Rice was doing a particularly clever experiment, in my estimation, and that was what we called male-limited evolution. So here, the paper published in Nature in 1996, Rice does this experiment. It looks horribly complex, and it is, and a lot of people puzzled over this diagram for long periods of time. But the action is all here in this main box. And what's happening here is males are put in Rice's experiment, about 800 males. They are going to compete with each other for access to a smaller number of females. And from that, he's going to harvest only the sons and chuck out the daughters each generation. Males competing, only males selected. The special genetics of the females allow these chromosomes to be transmitted from the father to the son without recombination. Hence, you can get whole genotypes to be transmitted without recombination. You can do selection among these different male genotypes for performance in competition for these females. The females are in the incubator. In the incubator, they're being pulled out every generation. And every generation that you do the male selection, you throw away the grandmaternal genes, so you prevent selection happening. So to make it simpler, this is what's happening here. The fathers are handing on haploid genotypes to their sons unrecombined each generation, and those genotypes are going to compete. Here's another portrayal of that. So here are the special clone-generated females, and this is exactly what's happening. This is what literally is happening. Sperm are being transmitted intact without recombination from fathers to sons each generation. So there you have a mercifully simple genome in terms of chromosomes. The big ones, there are 3 of them, compose about 99.5% of the genome, can easily be tracked together. You can pass them through using these special clone generator females. The females have X's that are attached together. It's called a double X or a compound X in the fly biz. And they also can carry a Y chromosome because of a particular deletion on one of the X chromosomes. So they're XXY. They're going to make eggs that carry 2 X's attached together, or a Y. When an X-carrying sperm fertilizes a Y-carrying egg, you get a son. That's part of the magic of the system. Sex chromosome transmission is backwards. And what it means is that big honking X chromosome, a huge component of the male genome, can be transmitted in the experiment to the sons, with the Y chromosome coming from mom. Okay, so this was a clever technique. It took me a while to figure out how it all worked, but I thought there might be some more utility to it. And here's the paper, the 96 paper. What Rice showed was that when males were allowed to adapt and females were not, females were held static in the experiment, and this was the focus of the work, that males improved in fitness and they improved in various components of mating and competition as sperm post-mating when more than one male mated. As a side effect of these adaptations, males became more harming to females. So another manifestation of sexual conflict perhaps, but it was never suggested that males were trying to harm females as some sort of punishment. In fact, what's happening is males are competing with other males. The more efficacious they are in post-copulatory sexual selection, the more toxic their ejaculate is to females. The females are the collateral damage in the equation. So this is interesting work. It helped to extend the phenotype of sexual conflict. Whoops. Sorry, it's a little dodgy on the range here. It helped to extend the phenotype of sexual conflict from the physical interaction of the sexes to what's going on in the ejaculate post-copulation. A whole bunch of seminal fluid proteins and peptides. We now know of well over 250 of them in the ejaculate of the male produced by this gigantic accessory gland. So this is an impressively complex cocktail of chemicals that influence the females in ways that you can undeniably say would be to the male's advantage, stimulating her to make more eggs, decreasing her attractiveness, literally changing the way she smells so that other males will be less attracted to her and she's less likely to be courted and to mate with somebody else, helping to release sperm from rival males from storage. The list goes on. And then there's the bad side effect. The longevity of the female is reduced. So these are male-male traits, male-male competition traits that have a side effect on females that results in this kind of conflict over mating involving the ejaculate. Okay, so it's pretty cool work. It was, again, happening in the lab when I joined in 1995, published in 96. But what I went to Santa Cruz to work on was this other kind of sexual conflict, the conflict over the genome. I've been working on things like stress resistance and longevity. Selecting on stress resistance meant primarily selecting on female survival of the stress because the males all died relatively rapidly because they're small. Turns out, selection on males is completely different under desiccation, for example, than it is on females because the males are all dead by the time selection is over and nobody mates under the stress. Selection on males is for fast development, early mating, maximum transfer of sperm, and assurance of their storage to be there for females when those females survive the stress. If they survive the stress, they're using stored sperm. It was holding up selection. And I got excited about this because I found if I freed the females from that male anchor, they could evolve even longer stress tolerance. And that's what I wanted to do with rice, was work on this particular problem. Okay, so what's the problem? Well, maybe there isn't one. If we look around nature, we know that the sexes are often really different from one another. It's called sexual dimorphism. We look at some species, we see that females outsize males, for example, in body size. We look at others, we see the reverse, males outsizing females. We see males that are ornate and females that are relatively less ornate. We see a lot of dimorphism in nature. What it tells us is that the genome has been under divergent selection in these species through perhaps macroevolutionary time, but also through microevolutionary time. And there's been selection to be different. And the genome has responded. It's found ways to make the sexes different. So why would there be a problem over the shared genome if selection is so good at making two different sexes? It can use different tools at its disposal. Hormones are an obvious one. Anyone can relate to the fact that the hormonal profiles of a female frequently differ from a male, and that leads to a different cascade of gene expression that makes females different from males on average. We've got various kinds of modifiers of the genome that can evolve. Duplications of genes where the female uses one and the male uses the other. It's a very common theme for solving this problem of the shared genome. So models of the evolution of sexual dimorphism assumed that there would be a problem between the sexes, a conflict, but that the genome could resolve it very quickly. So is there residual conflict? Is there conflict that remains to be resolved That's the question that I wanted to ask when I went to work with Bill Rice. Here's the kind of scenario that might be playing out. Let's take a species that is dimorphic. For example, Pecilia reticulata, the guppy. We know that males are ornate and females are plain, so our trait here might be coloration. We know that females are big and males are small, so this might be body size. Let's say it's body size. We have some kind of fitness optima. Males, we know from guppies, are selected for relatively small size, partly because they are much more preyed upon by big aggressive predators like Crenocicla alta, the pike cichlid. Okay? They'll eat them like popcorn, and they're highly visible because females make them produce these vibrant colors in order to gain mating success. So there's a conflict there. Okay, so body size, males, earlier maturation, smaller size. If we look at a real population of guppies, we will see that they range in size from being close to the fitness optimum, these females being relatively close to the fitness optimum, but some females being on this end. The supposition is that what's driving that is opposing selection on the two sexes, and that there's conflict that has not been resolved through the mechanisms that I've already mentioned. And so what's holding them back is what we call genetic correlations. And so this would be the case where large female, relatively fecund and successful, but because of genetic correlation, she produces large sons who are way down the fitness slope and less successful than smaller males. The sons of females, small females, who are relatively less successful in their female role, but are producing sons that are very successful as males in the next generation. That would be the scenario. That's the cartoon depiction of what's happening here. Okay, so I thought about this problem. I knew Rice was using this clone generating system, and I thought, why not use it to test this idea about residual conflict and sexual antagonism? So I did an experiment. I did an experiment with the clone gen system, a little bit different from Bill's experiments. I just wanted to survey, first of all, the genetic variation in male fitness. So taking what we called hemiclones, literally taking a sperm, and then copying it into hundreds of copies. Now I've got a whole population of what we call hemiclones carrying that same haploid genotype. I can test its fitness against hundreds of random genotypes from the same population. So it's heterozygous, it's not inbred, and I can test its additive genetic variation. Okay, if that means anything to you, that's a quantitative genetic term. It means its overall performance genetically. Okay, so there's a variance. It was astonishing, right? There's males in this population kept in the lab now for years and years that are half as fit as the fittest ones in the population. Why is the variation there? Well, part 2 of the experiment involves taking the exact same genotypes and testing them in the other sex. So there's the data. Looks like a mess, but believe me, there's actually a pattern here. So take a look, first of all, at the variance in females. The heritability for fitness, even higher than in males. The variance, even greater. That was a huge surprise. But what happens to particular genotypes? It's the same exact hemiclone that's being put into a daughter now instead of a son. So we can trace the fate here of, say, the second worst genotype in the population as a female. Well, she happens to be the second worst genotype in the population, or the second best genotype in the population as a male. Overall, the pattern is one of reversals. There's a lot of messiness in this. There's a lot of genes involved, about 18,000. So there's the pattern. I'll assert this happening on average, but you might be able to see that better if you look at it as a correlation. Now remember, the null hypothesis here is a one-to-one relationship. The same gene that makes a good father makes a good mom. The same gene that makes a good female makes a good male. So we would expect, under mutation selection balance, bad mutations are down here and they're affecting the sexes in a similar manner, and good genes are up here. That's our null hypothesis. What we found was actually the opposite, and that made it a whole lot easier to interpret. So what we found instead was this negative slope, strongly significantly negative slope, a whole lot of variation. Again, 18,000 genes involved in these hemiclones. There's a lot of things potentially going on. So there's a lot of noise, but the errors around these are actually relatively small. The heritabilities are quite good. And the pattern here is that If you're a genotype that makes a strong performing female, that makes a relatively poor performing male. Sexual antagonism, it's very difficult to interpret this pattern any other way. Okay, so there's our cartoon again of the relationship between the sexes for fitness, this time with flies instead of guppies. And if we were to do Rice's experiment all over again, selecting on males for performance in the male-limited design that he had used, and looking instead now at the females as well as the males, we might expect to see a shift in the distribution here as the males improve in fitness towards the male optimum. It's inevitable, but the big question is what happens with the females? So the supposition is, if we were to go and do Rice's experiment over again, taking away selection on the female, we would see a shift. And so what we would see is a movement of males towards their fitness optimum, maybe a little skinning up of the variation as selection takes away some of the poorer performing genotypes, but the females would move this way. We called this the gender load, the difference between the fitness peak and the realized fitness in the population. So the gender load, which my graduate students now tell me is not an appropriate term in today's society, but that's what we called it at the time. Maybe we should call it the sex load. The gender load, as we called it, was the difference between the optimum and the realized. And it was over here before, now it's over here, we've reduced it. It's the load in a population on one sex that's caused by the existence of the other. That negative consequences of having a high fitness dad for a female, for example. Okay, so this is what we would predict would happen if we did the Rice experiment. And so when I left Rice's lab and we published that paper on the intersexual genetic correlation, the crisscross pattern that I just showed you, the first thing I wanted to do when I arrived at Queen's was try Bill's experiment with selection and look for this pattern. Okay, so male-limited evolution, take 2. I promised you some wonderful Indian scientists would be involved in this talk. And when I set up my lab, there are various questions outstanding, and I wanted to do this experiment, and I wanted to recruit a postdoc to my lab. I advertised through email to various platforms, and I got a response from a professor in India, Dr. Amitabh Joshi. I knew Amitabh because he was a postdoc at Irvine when I was a graduate student, and I knew he was a very fine scientist. He was working in Larry Muller's lab, which was literally attached to Michael Rose's lab. He was working on density-dependent evolution. We were working on aging and other problems, but the 2 labs were merged into one. We had common lab meetings, and I knew Amitabh very well. And I trusted him, because I don't know if I would have hired Prasad based on the awkward phone call that we had that day that we actually arranged to call over a crackly landline between California and Bangalore, where he was finishing his PhD. But I trusted Amitabh, and GP came over. We had various questions in mind. We wanted to know the fate of the females in such an experiment. Did the female fitness go down? Rice had really not addressed this issue. We wanted to know about the fitness phenotype. Again, Rice did the experiment. He looked at a couple of traits related to sperm, and he looked at fitness, competitive fitness. There's no other phenotype. I was in his lab. I'm like, Bill, can we just weigh the flies? Can we send some samples to Mariana Wolfner at Cornell and look at the seminal fluids and find out what the magical stuff is that's causing these changes in sperm competition? No, fitness is what matters. I was very frustrated. I want a phenotype. I want to know what's happening, what it looks like. So this is why Prasad and I set off on this course. So we set off here, going back now, I promised you a talk over 3 decades, and here's the end of decade 1, 1995, now 2005. Prasad is in the lab. Prasad is doing his first talk to the department. Male fitness evolves at the cost of female fitness. Look at the date here. The date, come on, advance. The date, just highlighting that. It is almost exactly 20 years ago that Prasad did this talk in our department. And speaking of decadal milestones, here he is celebrating his 30th birthday in the lab with a Drosophila cake, of course. Okay, so Prasad helped design this experiment. We knew that if we were replicating someone else's work, like Bill Rice's work, and this paper was being cited all over the place, we're going to replicate it, we have to do it better. We have to do it with more animals, so bigger populations. We have to do it with more populations. He had 2 replicate pairs. We wanted 4 replicate pairs. We knew that if we found something different, we had to be able to point to it and say, our experiment is bigger and better. So 8 populations, 82 generations, a longer experiment, bigger population sizes by about 2-fold. Okay, and so we evolved them. We measure fitness of the sexes, if you're curious, through competitive assays. So these assays involve using marked recessive animals as competitors. So in the case of a female assay, we've got our recessive marked females, we've got our focal females. They could be the progeny produced from the male limited or the control type lines. And then we've got some males in there to provide the mating activity. And we've got something going on, protein that's limiting in the diet that they're competing for. In the male assay, a similar scenario, we put 1/3 focal to 2/3 marker flies. Using the recessive marked females, we can score paternity. So we have competition for mates in some kind of small cage structure, and we measure relative reproductive success compared to that marker, which is a vigorous recessive marked stock. Okay, the big result here was what we forecast would happen based upon the genetic correlations that have been measured in Santa Cruz, Santa Barbara, and the Rice Lab. That is, we saw a marked improvement in male fitness, so almost a 30% increase in male fitness over generations. It sort of flattened out after about 25 or 30 generations, but a corresponding loss of fitness for females. Female fitness for the same genotypes going down about 2/3 of a point for every point gained by males. It's a pretty strong negative correlation, and it's a pretty striking demonstration of the sexual conflict that we've seen through correlation with the experiments in Rice's lab, that spaghetti mess that I showed you. But maybe selection would allow us to characterize the phenotype better, because selection is going to take every piece of the phenotype that contributes to fitness and condense it in the population. Unlike the experiment I showed you, the quantitative genetic experiment, where you'd have that spaghetti mess all over the place, many, many genes, and probably a very difficult signal to measure and figure out exactly what's happening. So we measured various things in males: fitness, patterns of growth. Males grow more slowly. They go through they reach pupa at a later timing. They emerge smaller, so their growth rates are considerably lower than females. We found that the males had traits that made them more effective at courting and mating, as Rice had shown. We found higher activity levels of males, which seem to be related to fitness. That's also been shown in other experiments. And interestingly, we found an increase in the symmetry of these animals. Correspondingly, we found the lower fitness of the females, as I've already shown you. We found that females grew up like males, having delayed development and smaller body size. They became less attractive rather than more attractive. They had higher activity level, which in the lab environment at least seems to be detrimental to females, where it's advantageous to males. So we found a number of things changing. So we have something of a phenotype emerging, which was one of our goals. And we looked at the actual physical phenotype, for example, for growth. So males are smaller, males take longer, so their growth rate is slower. A question arises, why would that be advantageous? It's such a prevalent pattern when we look at invertebrates. They call it reverse sexual dimorphism. Reverse sexual dimorphism is such a silly term because it's the more common scenario in nature that females are bigger than males. It's just we are so mammal-centric that we think males should be bigger than females. Okay, so a third postdoc joined my lab. I think the picture of Stephanie Badum somehow blinked out of this. Stephanie Badum was involved in those experiments once we'd had them going. Jessica Abbott came to join us from Lund University. And Jessica had been working with Eric Svensson. Eric, I guess, was here a few years ago doing a similar thing that I'm doing right now as a guest of INSA. Jessica was an expert on geometric morphometrics. So things like Drosophila wings are wonderful for morphometric analysis. They're flat when you put them on a microscope slide, So they're easy to digitize. And in this particular experiment, she took the wings of male limited individuals and the wings of controls, measured both sides for a number of different landmark characteristics, and then you can put those through a geometric morphometric analysis that gives you various measurements of shape, size, and so on. So some examples of that. Here is one particular trait. This is just arbitrarily selected from over a dozen different traits describing wing shape, wing area, and so on. This is warp 3, our right warp 3 it's called. And what it describes is a sexually dimorphic difference between the sexes, as it turns out, in the location of a particular wing vein, the anterior vein. So take a look at a typical female here on the top, and then a typical male here on the bottom. Male's anterior wing vein is substantially inboard compared to females. Wing shape, you can see, is a little bit different too. So when you look at this characteristic, just to break it down, you can see that if you look at the more male-like pattern, which would be towards the bottom of the graph, we can compare female versus male. In the control, you can see that pattern. Females being like this, males being like that. There's the baseline dimorphism for the control. Now, male-limited lines have evolved in that direction of sexual dimorphism, becoming more male-like. So in the extreme, male-limited males here. But what's important too is that females have been carried along as well. Females are wing shape is genetically correlated with male wing shape. Selection has led to a more masculinized wing, but females are also carrying masculinized wings as a result of selection on the opposite sex. So we've got to change towards things being more male, and that was the general outcome from Jessica's work. Every single trait, except one, showed this pattern. And that one trait, come on, please advance. That one trait, whoops, was symmetry. Okay, if you take a look at symmetry, and you take a look at the control, you can see that the control, males are more symmetrical than females. Male-limited lines, males become super symmetrical. Smaller, slower growing, highly symmetrical. Maybe this is one of the advantages of so-called reverse sexual dimorphism, more careful development. And we know that symmetry is an important trait in sexual selection. Females evaluate males, among other things, on the symmetry of their traits, including their wings. So possibly this is what we call selection for perfection. An index known as fluctuating asymmetry was improved in the males. Interestingly, it became worse for females expressing male-limited genomes. They seem to run, when they run the male development program, development becomes less stable. So developmental stability is reduced for females expressing male-limited genomes. Okay, so everything that we measured, shape, size, wing loading, symmetry, all changed in the direction of extant sexual dimorphism for the males, and everything changed in the same direction for the females, except symmetry, a measurement where that would be a signal of increased instability in development. And if we noted at the time, you'll notice Jessica Abbott and Eric Svensson, authors on this paper. Jessica went on, among many, many other things that she's done in her illustrious career, to do some work on what are refraction patterns in the wings of Drosophila. For those of us that have done a lot of morphometry of things like insect wings, it's kind of annoying when somebody points out they're actually pretty colorful structures. The wings of a fruit fly, when you press them down and you make them intentionally high contrast against the background to make them black and white, like the image that I showed you before, when they're refracting light, you get these actual colors coming through that are quite vibrant. So if we'd known at the time, I'm sure we would have took a look at these wing interference patterns, because females respond very much to male wing colors. And just to condense a complicated slide down to a simple outcome, what do females like in a male wing? Magenta. That's the color. That's the technical name for the red that they love. And surely it also matters where the magenta colors are in that wing, in those compartments that are made up. That's what he's showing her. The peacock has shown Drosophila the way. He's showing her the wing. He's refracting colors to her as part of his displays. So pretty extraordinary. So from this experiment, we have some outcomes. We learned that morphological, developmental, physiological, behavioral characters, these were all implicated in the response. It's a comprehensive response. Again, we've got well over 10,000 genes potentially in conflict and potentially responding. We know that the X chromosome was particularly involved in the conflict, although we didn't do a lot to characterize the genes involved. And we know that characteristics that were under shared genetic control responded, but those traits that we found to be already male limited, including sperm, bizarrely, compared to Bill Rice's work, sperm competition did not change in our experiment. So we have that mystery. So take 3, another wonderful Indian scientist, or another pair of wonderful Indian scientists enters the picture, and that is Harshavardhan Thayagarajan. Okay, so take 3. This actually was in part a pandemic pivot. Harsha was working on a different project, a project which was simply untenable when things started to go towards pandemic lockdown. What we reasoned was a selection experiment could be considered to be essential maintenance, because if you don't do it, the lines will go extinct. We quickly started a selection experiment. We thought there were enough unanswered questions that if we do this Rice-style experiment one more time, we could move things in a new direction. We could ask, why was the outcome the way it was? What if we start from different populations? The world of sexual conflict in Drosophila, so many labs have their origin and their stock that they work on in Bill Rice's lab. I took it with me to Queen's. Jessica Abbott took it with her to Lund when she got a job at Lund. She took LHM to Sweden. Ted Morrow went through Bill Rice's lab. He took it with him. Max Reuter came to my lab. I went to his lab and trained his students and gave him populations from my lab. They spread all over the place. Like a disease, they spread all over the place. And everybody works on it. So Harsha and I, among other things, wondered, what's going on with other populations? So we started with 3 different populations from what had been worked on before. And here's Harsha, recently through and on to a postdoc in Switzerland in Thomas Flatt's lab. Wonderful postdoctoral opportunity. He's done extremely well. So the third Indian scientist that I said I would mention, he joined the lab as the pandemic circumstances waned and really cut his teeth working with Harsha and me on this male-limited evolution project. And that is Imran Syed. Imran is now just about done with his thesis, just about done with his writing and set to defend in 2026, sometime mid-year. So 3 great scientists. You can also see the ever-changing phenotype here of Harsha. Facial hair, no facial hair but long hair. Yeah, a chameleon. Okay, and here he is at work in the lab. Okay, those populations came from Massachusetts originally. They are the populations that I worked on, some of them in Michael Rose's lab. If you're tomorrow at the my mini talk on aging, the lab talk, I'll talk more about this phylogeny. The important point is we took 3 populations from this phylogeny. Harsha selected the odd-numbered ones just to be awkward and annoying. We worked on these 3 populations separated by several thousand generations of selection in the lab. So we thought that was a good way to achieve independence and replication. We got the first result. Different populations, they all did something that had been done by LHM before. Just like LHM, we saw a substantial increase in male fitness. So here we test them the same way that Bill Rice tested them. We see an improvement in fitness going by about 30% once again, and it's consistent across lines, ML versus control, ML versus control, ML versus control. Right, so this was a big encouragement, so we were happy with that result. We test the females. Here are a couple of different assays shown where the females now, from the ML lines versus controls, you can see that the female fitness shows the opposite pattern. We've got fitness of females reduced, significantly reduced, again, consistent with the observation of sexually antagonistic evolution. Right on. Things are going really well. We are totally stoked about this, and we're thinking, Okay, we're getting the collaborations lined up to do the sequencing, to do the gene expression work. We're going to take the phenotype down now to the genotype. We're really excited about this. These are testing with 2 different females. So these are testing with wild-type females, where the males would not have adapted to them, like in the rice experiment. These are testing with females, the clone generator females, that they might have adapted to, as rice had suggested. The important thing is that we see a generic improvement in male performance, and it does not matter which females they were competing for. Contra Rice's take and narrative on the outcome of his experiment, he thought it had to be adaptation to the females. What we've shown is that it is actually an adaptation of the males that is innate, and that their performance is improved, and that it does not matter what kind of female they were competing for. They improved, and their fitness had increased. So for a completely different reason than Rice had suggested. Okay, and then what I promised last time, the female fitness has gone down. You can see it's a little variable from replicate to replicate, but it's significantly reduced. Good, okay, that's happy. Female fitness declined, male fitness went up. Male fitness was improved in the absence of the special females used to evolve them. Everything is looking great until we do the experiment completely naked. No genetic constructs, no weird genetics to make the males, and the fitness differences almost completely go away. They're there in some experiments, but they're slight. They're there in one experiment that Harsha did quite substantially using the technique we call the hemiclone technique. And that's in the paper we recently published. So there are some results that show male fitness has improved, but it's kind of a slippery character. And so we start thinking about other possibilities for adaptation by these males. When you look at that clone generator female, whoops, again, you've got characteristics here like the reverse transmission of the sex chromosomes, females giving the Y, males giving the X. That also means that females are donating cytoplasm through the egg. And these females, these double X females, they come from a stock center. They were actually derived by Hermann Muller himself, Mr. Ratchet, Dr. Ratchet, back in the 1940s. And so potentially quite different for things like their mitochondria. So we start thinking about why males would be so sensitive to the genetic conditions. And we've got some candidates here, the foreign cytoplasm. We've got the translocated chromosomes that were used in the females. We've got the reverse inheritance of the Y, reverse inheritance of the X. These candidates for why our results are so inconsistent and difficult to reproduce when we raise the males under different conditions. And actually, as I was thinking about this talk just last night at about 3 o'clock in the morning when I woke up thinking about this talk, I realized there should be a 5th spoke on this. And I think it explains why Harsha's results are different with the so-called hemiclone system than with the experiments that we did on fitness. In those experiments that I just showed you, we allowed the genotypes to mix randomly in the MLs. In the hemiclone experiment, the X was kept together with the other major chromosomes in the genome. I am now thinking this may be the most parsimonious explanation for the disappearance of the male advantage. The effect is based on gene-gene interactions across X and other chromosomes. And that would make perfect sense, explaining why it disappears when we scramble genotypes. But something I'm going to talk to Harsha about ASAP. Okay, so we're trying to figure this out. And right now, I'm just going to leave it at it's complicated. And evolution is relentless. Evolution is changing. It is clearly operating on some of these artifacts, but in different ways in different replicates. In 2 populations, reverse transmission of the Y chromosome really matters. In one, it matters not a whit. In one population, it really matters which cytoplasm was used. In the other 2, it doesn't. So we've seen evolution happening in these replicates in different ways, grasping at different ways to adapt, which is the theme of a paper that we're working on right now. It's a more complicated outcome, but it raises some really interesting questions about co-evolution in the genome. Okay, so I'm going to wrap things up here by sort of pulling together these results in a way that I hope convinces you that it's more interesting than just a fruit fly thing. And that's the way it was 20 years ago when I was, first talking about this and giving talks on sexual conflict. Is it just a fruit fly thing? Well, since that time, there have been a whole lot of studies, and this is just a handful of them to give you an idea of the diversity. We've got studies in other insects, we've got studies in reptiles, we've got studies in birds, we've got studies in big mammals, we've got studies in the lab, we've got studies in the field. Intralocus conflict, this conflict over shared genes, seems to be a pervasive thing. And 20 years ago, when Joran Arnquist and Locke Rowe wrote the book on sexual conflict, this form of conflict occupied all of 4 pages in a 290-something page book, a couple of percent. And they said, based on the work of Chippindale and Rice, maybe intralocus conflict is something that could be important, but at this point, we just don't know how common and consequential it is. I would submit that the last 2 decades have suggested that this kind of conflict is common. In fact, it's everywhere, and it is consequential. It may even be impacting traits in our own species. So some work that started with Campiriano and Ciani's work back in 2004, a whole bunch of follow-up papers, including reviews by Rice and Gavrilets, establishing that intralocus sexual conflict could be playing a key role in our gender diversity and in our sexual preferences, helping to explain perhaps why we have such a rich diversity of sexual preferences and gender in our species. Here's an example of something that's relatively easy to understand, a conflict over testosterone. Beautiful work by Suzanne Mills. She selects on testosterone. High T lines, good for males, bad for females. Low T lines, good for females, bad for males. The classic crisscross design for a particular trait that seems to be easy to understand, an androgenizing hormone being beneficial to the sex, the male sex, but not the female sex. How about the plant world? Beautiful work by Linda Delft. I won't show you her data, they're kind of complicated to portray, but basically in Silene latifolia, you've got a system where female flowers are much larger than male flowers. What Delft's work shows is that the conflict here is over size versus numbers. Females benefit from having relatively few large flowers. They want to have an inflorescence and they want to make lots of seeds. In females, the number of seeds is correlated with the size of the flower. The calyx of the flower correlates with the number of seeds in the calyx. The size of the calyx does not correlate with the amount of pollen in the anthers. So to make lots of pollen, males have to make lots of flowers. To make lots of flowers, they make small flowers. It's incredibly inefficient. And so we see a conflict between size and number of flowers. In Silene, Delft selects upon it. She finds fitness disadvantages to males with large flowers. You see other traits carrying along. Here's a figure that Linda sent me recently, showing the difference between 2 different males after they've begun flowering. This is a high fitness male. This is a male that, after flowering for a month, is a complete burnout, has produced all kinds of small flowers, and he is now withered and dead. He has thin, flimsy leaves. This female-like male has thick, waxy leaves. He's going strong, but he has not been as successful in pollinating other Silene individuals. Here's one of my favorites. This is the clubwing manakin. The clubwing manakin is a dweller of the Ecuadorian rainforests. They call it a clubwing manakin because of this massively ossified ulna bone. Okay, so let me show you that cool animation again. There we go. Take a look at this ulna bone. This is the only bird in the world that doesn't sing with its mouth, with its beak. It sings with its wings like a cricket sings with its legs. It's called stridulation. The males sing to the females by vibrating their wings together over their heads. These massively ossified ulnas rub together to make a hurting sound that the females go crazy for. Okay? Now, you can imagine if you have a highly ossified, knobbly bone like this that is completely filled, as opposed to being hollow in close relatives in other species of manakins, that's going to be a bit of a downer in terms of flight performance. So, sexual conflict, how is that? Well, females may be driving this, but they're also paying a price for it. So here's a white-cap mannequin. Here's its hollow, gracile, all in the bone. Everything's cool. Now take a look at the clubwing mannequin. Here's the male with that big, knobbly, solid bone that he's going to use to stridulate and sing. But that's the female. She doesn't need to sing. She doesn't need a big, knobbly bone. She is suffering the fitness consequences of carrying around the sexually selected trait that has been selected in the male. Conflict in action. Our own species, again, I mentioned gender diversity. How about stature? Here's a paper from Sanjak et al. Looking at the UK Biobank study that spans 3 generations in the UK and extensive full genome data set, full genome sequences for a huge number of individuals, all kinds of other data collected. This paper, one of the things they emphasize is the sexual conflict that's being expressed over stature. Males in real populations in the UK look like this. These looks almost like cartoon data that I showed you earlier from my cartoon for fitness functions. These are the fitness functions. Modern humans, birth control doesn't matter. Males are shorter than the fitness optimum. Males that are about 6'1" have more kids than males that are more like 5'9", like the average in the population. Females are too tall. Shorter females have more offspring. In the UK, in the last 3 generations, these are real data, this is real selection happening, this is real sexual conflict, and this is modern Homo sapiens. What about other traits in our species? Well, Bernie Crespi thinks that our brains may be a battleground in terms of sexual conflict. Why? Because our brains are the most imprinted parts of our entire genome. Our brains are full of hundreds of genes which are switched off parentally by one sex but not the other. It is a battle over gene expression and growth, and the conduit of that battle is the placenta and the growth of the fetus. This is a kind of sexual conflict that was pointed out by Robert Trivers in the 1970s, a conflict over growth, parent-offspring conflict, male-offspring conflict over the fetus. Crespi and colleagues think that this is what's happening. Conflict over brain growth leads to undergrowth and overgrowth of certain parts of the gene of the brain during development, leading to known psychosocial disorders such as psychosis and autism. That's their hypothesis. They've got heaps of data to back this up, encyclopedic catalogs of imprinted genes and their phenotypic effects. It's pretty cool, but this could be a battleground that affects how we think and how we interact with our colleagues around us. Okay, just to sum things up. Every inventor dreams of inventing the perfect machine. In the 20th century, one of those perfect machines was the flying car. A flying car would let you cruise on the road, but when traffic got tough, you could lift off and fly above your neighbors that could not afford the flying car. No compromises here. It's a luxury mobile, it's a great cruiser, and it's a great flyer. Well, what is the most successful flying car of all time? It happens to be the Moult-Taylor Aero car. A few hundred of these were produced around the middle of the 20th century by a guy named Moult-Taylor in Seattle. It flies okay. It flies okay. It drives okay, but you don't have much space, you don't have much power, and certainly not great if you have to tow the wings around behind you. So it's very hard to make 2 machines that do things well at the same time when there are trade-offs between them. Motor vehicles happen to be one of them. Have inventors solved the riddle of the flying car? Well, it looks like it may be. This is the Terrafugia TFX. The Terrafugia TFX is that car. It is the dream come true. And hundreds of people shelled out hundreds of thousands of dollars in advance to reserve their copy of a Terrafugia TFX, only to find the company bankrupt itself and disappear. Did Terrafugia ever make a flying car? They had to do something. And there it is, Terrafugia's flying car. I would submit Terrafugia's flying car does not look a whole lot more practical than the Molt-Taylor error car did. It's challenged, it's hard. Making two different things out of one common set of parts that do things well is difficult. Okay, so I've talked about two different kinds of conflict. On the bigger scale of things, this arms race kind of evolution between the sexes has been suggested as being an engine of reproduction, an engine of speciation, sorry. That is, the two sexes are going to evolve relentlessly in connection with one another over control of reproduction. And the traits that are involved are going to be obviously reproductive traits, signaling traits, things that if they were separated in allopatry on two islands that were identical in every way, shape, and form, they might still diverge by chance to become different and then to not breed with each other when they come back into contact, hence the speciation angle. The arm wrestle or the tug of war, this is more like a capacitor for variation. The sexes are not at their optima, the sexes are compromises, and there's a blur between them. There's a spectrum, a rainbow, what do you want to call it? Some of these characteristics that we see that make our species so diverse and so wonderful and so interesting, they might be there because of intralocus sexual conflict, the conflict over shared genes, not making two perfectly dimorphic types in the population. And I think that's pretty cool. So Henry Kissinger famously said that nobody will ever win the battle of the sexes. There's just too much fraternizing with the enemy. And I would submit that that's true of the arms race, and that's true of the arm wrestle. Not even the super male is going to be able to overcome those odds. So thank you very much. And I'll let you have questions if you do.
Hey, thanks so much. That was a fantastic talk. So, if I understand this correctly, if one of the two sexes is under a stronger selection pressure, you expect there to be less dimorphism. Is that correct?
If one of the sexes is under
is under a stronger selection pressure, like in your experiments, you expect dimorphism to be lower. Is that?
Well, actually, what we see is the shift in dimorphism in the direction of one of the sexes with the other sex towed along. So our evidence is more like a shift where the dimorphism itself wasn't necessarily changed. In fact, it was remarkably stable for morphological characters. It's just the animals became more male-like and the females became more male-like, but the dimorphism itself was not collapsed.
So I guess my question is related to that. So why does that happen? Because I would expect that the sex that's not under selection you should just start to see a greater spread in that sex, or is that?
This is a problem with the experimental design. If you do sex-limited selection and there are sex-limited genes, they are in free fall. So that effect is something we were cognizant of. We knew the experiment couldn't go on too long because genetic drift on female-specific genes would eventually lead to the decline in female fitness.
Sure.
We've done various things to try and test for that. For example, crossing lines to make heterozygotes.
Yeah.
Because then you would eliminate, presumably there'd be different mutations that accumulate in the different replicates. And we've seen relatively little apparent contribution of that over these time scales.
I see. So, but if one looks at natural populations, so let's say species where you have high degree of dimorphism versus those where you don't, can one sort of come up with a theory for why certain species, the sexes of that species diversified, whereas in other cases, things
Why are
didn't.
some sexes more monomorphic and some
Yeah.
highly diverged?
Yeah.
The differences in cross taxa in the lability of sexual dimorphism are extraordinary. And I showed some real highlights there. Species where males are absolutely miniaturized and females are gigantic.
Sure.
What allows them to evolve such radical dimorphism, whereas other species seem to be stuck with unresolved dimorphism? I don't know. I mean, evolutionary age might be something, but I don't think that really applies to a fruit fly.
Yeah.
And we've seen evidence that there's some pretty abundant unresolved conflict in D. melanogaster. So I don't have a great answer for that other than historical contingencies and breakthroughs that allowed big differences to evolve in some species, but not others.
Not others. Sure.
Okay.
So,
So I'm not from a biology background, so pardon me if I don't use right language. But most of the experiment you showed, each of them had male selection. Are there experiments about female selection, and why you particularly chose male selection? And are the results for female selection similar?
Yeah, why do we do male limited and not female limited evolution? The answer to that, I may have glossed over it in my talk. The answer to that is that there's no chiasma formation in male Drosophila, so they don't have crossing over. So it is much easier for us to transmit unrecombined chromosomes in males from father to son because they don't cross over. So all we have to do is keep track of the chromosomes and we can do the experiment. To do female limited evolution is more challenging because you have to suppress recombination to do the same thing. Now that's possible for parts of the genome in Drosophila, very difficult for the entire genome. Where
But
it's been done,
could it change the results if the mechanism for females is different?
I mean, it could definitely change the results. In my imagination, no, that would be, obviously we get the exact flip side of this if we did female limited evolution. We have done a female limited evolution experiment that went with Jessica back to Sweden. It's been done on the X chromosome only. So it's part of the genome that she took and used a recombination suppressing system, what's called a balancer chromosome. In her work on female limited evolution, it shows some of the same responses, but female benefit, male detriment genes elevating in frequency. They also find evidence for some contributions of compensatory selection. For example, using the balancer chromosome and the weird markers that it has, there's been evolution and adaptation to that, as I showed in our experiments. When you do these weird genetic manipulations, you have to be prepared that your lines are going to change in response to them. They're going to adapt. But yeah, no, the female limited evolution experiment, I would have done that in a heartbeat if it was straightforward to do with the same tools.
But with other species, is it done?
With other species, perhaps. Now, even with Drosophila, there is a different way to do a sex-limited evolution experiment, and that is what's called the middle-class neighborhood approach. Max Reuter, who I mentioned in one of my slides, has a female-limited evolution experiment going with Drosophila right now, yet to be published. But what they do is they standardize the reproductive success of one sex. This is the riff here is middle-class neighborhood. Everyone has 2 children. Okay, so the middle-class neighborhood design, if one sex is always made to only have 2 kids, then it only replaces itself in the population, and now there's no selection on that sex. And then you allow the other sex to have as many offspring as it can through selection. So the female limited experiment, you'd be using markers and collecting only 2 offspring from each male, but you'd allow any given female to have as many offspring as she can produce. So it can be done. There are a whole raft of technical issues again that make it not a perfect experiment. And I think that was one of my lessons today. That said, we've got work going now on nuclear mitochondrial co-adaptation that's looking really, really interesting, and it's because we found that artifact in our experiment. Yeah. Which is actually a form of sexual conflict, because mitochondria are female transmitted, and they have female fitness in their lines.
this is a little memento for you to remember this talk.
That's so cool. That's so cool. IISER Pune plaque. This is really beautiful. Come up and take a look at it. This is really nicely done. Thank you. Thank you very much. Yeah, it's an honor to be here.
Yeah.
Gift is. Thank
Thank you. And I need to thank Indian National Science Academy who made this possible, IISER Pune of course, and finally all of you for turning up.